So yes, good evening, everyone. This side Dr. Abhijeet, educator, biochemistry at Auricle, and today I will be discussing a very important topic that is the biomolecules and cells. So we have started this 21 days revision series program, especially for the ones who are going to appear for the prof exams, right? So I will be discussing the topic biomolecules and cells. Over here, guys, I have taken the most important questions, right? The previously asked questions which are being asked like repeated number of times, right? In various university exams across India. I will be revising the entire topic in the form of Q&A, that is the question and answer format, right? I will be telling you how to write the answer, how to approach the question, how much time you should spend on like different questions, right? I will be telling you the key points that you have to include while writing the answer, right? So everything I will be covering along with the question, okay. So yes, the ones who are going to appear for the internal exams, the prof exams, it is beneficial for them. But the ones who are preparing for NEET PG, INI-CET or FMG exam, they can also benefit from this particular lecture, right? Now, before starting, guys, the questions that I will be covering up today, that is the major biomolecules, right? That is the carbohydrates, lipids, proteins, and nucleic acid, the basic structure I will be discussing along with the function. I will be discussing the amino acids, the different levels of no protein structure, the primary, secondary, tertiary, and quaternary structures, along with some lipid molecules, right? So that is will be the topic of our discussion for today, right? So the long essays, basically, they are usually asked in the form of 10 marks question. The short essays, either they can be like 8 marks or majority of them will be 5 marks questions. And short answers are either 3 marks or 2 marks, right? So I will be telling you how much content you have to write in each of the following answers. And yes, of course, the topic will be entirely covered in that particular answer only. Now, guys, little bit about the Oracle app. You already know for flexible learning, guys, if you're not watching the no live sessions, right, you can always go on for the recorded videos that are available on the app. Secondly, we are conducting the live classes daily from 6 p.m. to 10 p.m., right? On the YouTube channel also and also on the app. The various practicals, for anatomy, biochemistry, and physiology, even for the internal exams, the practical sessions are being recorded, right? We have a recorded version also. You can always go on for those practical videos before going on for the exams. Now, just 1 month before the exams, you guys keep on asking us like what to do. We cannot read the entire book. We cannot go on through with each and every topic. Guys, remember just before the exam, please go on for this under 20 hours revision series. In this particular video, in this 20 hours, we have covered the entire important, potent questions that can be asked in your exams, right? Even the most important one, that is the hard copy book set that is now available at your doorstep. You can easily order them, right? And secondly, we have no included, we have no decrease your burden, guys, of no like going all through the difficult textbooks, right? So you can just go on for these notes, basically the book set. Basically, in this, you will have the Q&A format, that is the question and answer format. Like the entire question topic will be discussed in the form of Q&A, whether it is a long question or a short question, right? Even the MCQs are also discussed, right? We have fun zones also. We have like schematic diagrams also. So everything is being combined and it is present over here. Foundational classes, guys, I've already told about this, right? The live classes, we are scheduling it daily from 6 p.m. to 8 p.m. I mean 10 p.m. daily live on the app, right? Apart from that, we have the express exam module where we cover everything under 20 hours, right? One-to-one doubt clearing session. If you have any doubts, guys, you can just book an appointment prior, okay, and with the team, with the management, and you can always get your doubts cleared with the educator, respected educator, right? The hard copy book set, I've already told you, it's being basically curated by the team. And yes, so it has no answer writing parts also, right? Even the concepts are being explained in the form of Q&A and everything. The question bank, guys, after each and every topic, you will be having basic to advanced questions. All the questions are being covered, guys, right? about the last previous year topics, the previous year questions, right, of various national exams like NEET PG, INI-CET, or FMG exam, right? Even the new pattern questions now, the clinical vignettes and all those are created by the expert team. They are also being over here, like included over here, right? So yes, you can always go on for these, no, solving these MCQs after you read each and every topic. You can just go on for these also, right? So the practical sessions I've already told you, they are also available. And the basic subscription plan, the foundational plan, guys, that is, yes, you can go on for the foundation plan, the advanced learning plan, right? The complete learning plan, and we have the ultimate MBBS plan, ultimate MBBS plan. So you can use my code Dr. Abhijeet 20 for no additional discounts, right? Now, before no starting up with the question, guys, two, three things that I have to include over here. Firstly, guys, try to attempt all the questions in the exam. Attempt all questions in the exam. Very, very, very important. Right? Attempt all questions. Whether it is a long answer question, whether it's a short essay question, whether they are MCQs, you have to attempt all these questions. Now, guys, try to give like more time to the long answer questions, right? Then to the short answer questions and MCQs, right? Time management is again very, very, very important, guys. Time management is very, very important. Try to start with question number one only. It's no, they have a different saying. No, we can start from the last, up to you guys. The more like you're confident about any question, just start with that. It will no boost up your confidence throughout the exam also. Right? The question which you know first, just try to attempt it first, right? So just try to spend minutes on some 5 to 10 minutes on long questions, and then no, try to decrease the time according to the marks of the questions allotted, right? Now, apart from that, guys, I always ask you to draw the structures. Very, very, very important. Draw the structures. Do not write the answers in paragraph form. No examiner will be no reading your paragraphs. No one is interested. Try to write in points. Very, very important. Highlight the key words. I will be like telling you how to do it. Okay. Try to write in points. Try to highlight the keywords. Okay. Draw the structures. Again, I'm not telling you to be no draw something very fancy. You have to just draw the schematic structures, right? The schematic line diagrams are even like important. The schematic structures are important, right? Try to draw flow charts. Very, very important. Flow charts. Again, if anyone has written the whole answer in a paragraph form, we know out of 10, we usually tend to give 6 or 7 marks. But if you have included flow charts or no keywords, no highlighted and in the points, we like give 9 or 10 also, right? So it depends on how you are actually writing the answers, right? So that is something that you have to keep in mind while starting the paper with, right? So yes, flowcharts are again very, very, very important, guys, right? Reactions are important. If there is any reactions that we are discussing, they have to be like specified. Reactions, basically, you have to write the enzyme. Okay, you have to write the ions. Okay, you have to write okay, the enzyme, the ions, everything you have to just not the substrate and the product. You have to include the enzyme, you have to include the ions. So you have to like draw it very nicely, guys, right? So that is something very important. Now, let's start with the very first question. Let's start with the very first question. Now the question says describe the structure and function of the four major type of biomolecules. So this is question number one, and that too for 10 marks. Now, guys, there are four biomolecules that are being given in the question: carbohydrate, lipids, proteins, and nucleic acids, right? So remember, guys, you have to take every important word of the question into your answer. You cannot tend to skip any important keyword from the question. Okay, so the question says carbohydrate, lipids, proteins, and nucleic acids. So in the answer, you have to cover all the four. Okay, it's not like you'll just cover the three and you'll left the fourth one. Absolutely not. Whatever is there, the structure and function. Please read the question. You have to tell the structure also and function also. So yes. Now let us just start with the first one. That is the carbohydrates. Carbohydrates. Now, when I say carbohydrates are nothing but the hydrates of carbon. Okay, they are nothing but the hydrates of carbon. Okay, or simply, they are organic compounds. Organic compounds, which you already know, they are made up of carbon, hydrogen, and oxygen. Hydrates of carbon, that is carbon, hydrogen, and oxygen, right? Or one more definition that you can write, that they are made up of, they are aldehyde or keto derivatives. They are aldehyde or keto derivatives of polyhydroxy alcohol. Very, very important. Polyhydroxy alcohol. Now, this is something which you have to remember. They are aldehyde or keto derivatives of polyhydroxy alcohol. Now, this is the actual definition of carbohydrate. Now, glycerol. Just if you can draw the structure, it is again very, very nice, guys. So remember, guys, glycerol, you already know it has 3 carbons and 1 carbon. It basically, no, the valency of like carbon, it is 4. So it will be like forming 4 bonds, right? So obviously we have polyhydroxy. So to all the carbon, 1, 2, and 3 carbon, basically OH is attached. OH is attached. Polyhydroxy, OH is attached to all the 3 carbons, and 1 carbon will be like making 4 bonds, obviously. So yes, at first we have to balance it by hydrogen, second also, and third also. So this is the structure of a glycerol. Now from this glycerol, basically we are making aldehyde or keto derivatives. Aldehyde, guys, you already know the functional group, it is CHO, and keto derivative, it is C double bond O. Okay, that is like we have to make it from here. Now, guys, you can just remember if we try to remove hydrogen from the first carbon over here, right? 2 hydrogen from the first carbon. Obviously, the first carbon will be left with oxygen and 1 single hydrogen, right? So that is again CHO, right? CHO, like this it will be left. If we are just removing hydrogen from the first carbon, rest the entire structure will be same. Rest the entire structure will be same, right? Now this structure, it is called as glyceraldehyde. Glyceraldehyde. Again makes sense. This is glycerol. And you are adding aldehyde group over here just by removing H2. So it is glyceraldehyde. Now this is something which is called as the parent carbohydrate. This is called as the parent carbohydrate. Right? This is called as the parent carbohydrate. Now, again taking a situation where I'm removing the H2 from the second carbon, right? So yes, the first one will remain the same. If we are removing the hydrogen, both the hydrogen we are removing from the second carbon, it will be left with oxygen, and oxygen will form double bond just to satisfy the valency of the second carbon, because the carbon forms 4 bonds. Then the third also will remain the same, right? Now, guys, if you can just see over here, this structure you already know, guys. This structure is of acetone. Acetone. Yes, CH, C double bond O, CH2. Now, yeah, this will be CH2. CH2, C double bond O, CH2. And 2 hydroxy groups are added on first and third carbon. So it is called as dihydroxyacetone. Dihydroxyacetone. It is also a carbohydrate. It is also a carbohydrate, right? We have dihydroxy, 2-hydroxy, and acetone. It's also a carbohydrate. So it's a keto derivative. Dihydroxyacetone is a keto derivative. And glyceraldehyde is an, yes, aldehyde derivative. Aldehyde derivative of polyhydroxy alcohol. This is how basically the structure looks like, right? Now the general formula, guys, you already know the carbohydrates, they are being classified into many types. The carbohydrates are being classified into many types, and you already know we have monosaccharides, we have disaccharides, we have oligosaccharides, and we have polysaccharides. So again, the basic definition, guys, you already know. Mono means one single sugar unit, And again, yes, the structure is important. I mean, the basic formula for monosaccharides, it is CnH2O. N basically represents the number of carbon atoms. N is equal to number of carbon atoms. Very, very important. Number of carbon atoms, right? Disaccharides, you already know we have two potential sugar units. Oligosaccharides from 3 to 10, and more than 10 we have polysaccharides, right? More than 10 we have polysaccharides. Again, over here, just try to write it in very, very short, like monosaccharides, one single sugar unit, disaccharide, two potential sugar units. You already know the disaccharides, right? For example, monosaccharides, you already know, right? Monosaccharides, you already know we have glucose, we have fructose, right? These are monosaccharides. Disaccharides, we have already discussed earlier also, for example, like sucrose. Sucrose is basically composed of glucose plus fructose. We have two potential sugar units. Or lactose, that is glucose plus galactose, right? Polysaccharides, okay, you can just go on for the examples like starch, glycogen, okay, or cellulose that we have discussed in the normal classes, or cellulose, right? So basically, this is the structure of the carbohydrate, how the carbohydrates are formed, and the basic classification, right? Now they have asked a function of carbohydrates also, and I think this is the easiest one. The function of carbohydrates. The function of carbohydrates. Firstly, you already know the energy. It is the energy source, right, guys? They are the primary source of energy, and especially if we talk about the glucose, right? Glucose, it is a monosaccharide. Okay? So it is used by the cells to generate something called as ATP, adenosine triphosphate, that is called as the energy currency of the cell. It is called as the energy currency of the cell. Powerhouse of the cell is mitochondria. Secondly, you have to remember energy storage, that is basically in the form of these polysaccharides, right? Energy storage. For example, if we talk about glycogen. Now, in the well-fed state, guys, now when I say well-fed state, now well-fed state is some like you have consumed the food. Okay, right now you have eaten your dinner, right? So there will be enough glucose in the body. There will be enough glucose in the body. Some will go for glycolysis to generate ATP, while the rest glucose, guys, will go into the liver, and they will be stored in the liver. They will be combining with each other. The glucose will be joining with each other to form glycogen. Okay, that will be glycogenesis. Genesis means formation of glycogen. So that after 4 hours, when there is depletion of glucose in the body, the liver will break down this glycogen and it will release the glucose unit so that it can meet the requirements of the glucose in the body. Right? So the second most important function, it is the energy storage, right? Even It also helps in cell signaling. Cell signaling. Now, you already know on the surface of the carbohydrates. Now, if we just talk about the cell, and you already know the cell is covered by a membrane that is called as cell membrane. And on the cell membrane, guys, on the cell membrane, we have certain carbohydrates which are present. Okay, there are certain carbohydrates which are present. Okay, these are nothing but glycoproteins, or they can be either glycolipids. Okay, the glycolipids. Okay, they are having an important role in cell recognition, and yes, the cell recognition and communication, right? So yes, that is something that you have to write about carbohydrates. The second part that they have included in the question, it is the lipids. Lipids. So lipids. The second one, that is the lipids. Now, now they are basically the molecules that are they are hydrophobic molecules. Now, they can dissolve in non-polar solvents. Now, when I say non-polar solvents, guys, you already know basically I'm talking about ether. But as I said that they are hydrophobic molecules, right? So remember they dissolve in non-polar solvent, but not in polar solvents like water. But not in polar solvents like water, right? So that is something you have to remember. They are not they basically like they are similar in structures, but obviously they are non or they do not dissolve in like polar solvents like water, but they can dissolve in non-polar solvents such as ether or chloroform, right? So remember basically we have different no lipids in our body, right? We have simple lipids, we have simple lipids, we have compound lipids, we have compound lipids or complex lipids, we have derived lipids, we have miscellaneous lipids, miscellaneous lipids, right? Now, when I say simple lipids, basically I'm talking about the normal lipids, right? They are basically formed from the fatty acids and glycerol, fatty acids and glycerol. For example, right, the triacyl glycerol, the triglycerides, the triglyceride. Compound lipids, we have fatty acids plus glycerol, plus we have some other no components like phosphate. So it will be phospholipid, right? If the other component is no carbohydrate, it is fatty acids plus glycerol plus carbohydrate, it is glycolipids. If the other component is sulfate, then it is sulfolipids, right? So we have like compound lipids, simple lipids, derived lipids, and miscellaneous lipids, right? Derived lipids, they are again derived from triacylglycerol. The triacylglycerol will be broken down by the by hydrolysis into fatty acids and glycerol, right? And some miscellaneous lipids. So you have to just remember about this, right? So these lipids are also know they are composed of carbon, hydrogen, and oxygen, right? They are also composed of carbon, hydrogen, and oxygen, right? So yes, the function of the lipids that you have to remember over here, the functions of the lipids, firstly, it is the energy storage. Again, the same function as yes, the carbohydrates, we have the energy storage. And you already know, basically, I am talking about the triglycerides, right? So basically, they store no energy in the form in the adipose tissue. in us. Now, I have talked about phospholipids. Secondly, you should know that the phospholipids are basically the important component of the cell membrane structure. Important component of the cell membrane structure, right? The cell membrane, bilayer phospholipid, right? The cell membrane structure. So yes, remember guys, it is also responsible for protection, okay, insulation. Apart from that, they are also responsible for production of hormones. That is something very, very important. So, for example, like you already know the steroids like cholesterol, right? Cholesterol, okay. So the cholesterol is basically a precursor. It can produce hormones like testosterone, estrogen, even cortisol. So basically, the lipids are also helping or involving in the function of production of hormones also, right? So remember, yeah, these are the most important ones that you can include in the answer, right? Again, I'm discussing the entire one. So yes, you have to be very choosy. No, you have to write exactly the correct ones. I mean, if you have, for example, lipids also perform signaling function, right? So such for example, like icosanoids, right? If you don't know about that, don't mention it over here. Like if you have a complete knowledge on someone, just try to include that part in the answer. The third one, basically, we have this the proteins. The proteins. Now, the proteins, guys. The third one, that is the proteins. Now, proteins is something that you guys already know, right? Proteins are basically the large molecules. They are composed of amino acids. They are composed of amino acids, right? They are basically the amino acids are linked by peptide bonds. I will be discussing the peptide bonds in detail today. So remember, they are linked together by peptide bonds to amino acid A1 and amino acid A2. Basically, they are joined together by the removal of water molecule. And yes, we have peptide bond between them, right? Now, if we talk about the structure of, yes, if we talk about the structure of, yeah, the proteins, right? So now, the proteins have basically four levels of structures. I will be discussing in detail all the four levels in the upcoming questions. So, but over here, remember, we have four levels of structure. We have the primary structure of proteins, the secondary structure, the tertiary structure of proteins, and the quaternary structure of proteins, right? So primary structure is nothing but, in short, guys, you have to remember, it is the linear sequence of amino acids. It is the linear sequence of amino acids, right? Secondly, remember, yeah, the secondary, the amino acid starts folding, right? So it is local folding. It is the local folding. And it will result in two main structure. We have alpha helix and beta pillated sheath, just like the sheaths or the plates of a no sari. So we have the beta pleated sheaths, right? Tertiary, you already know, it is the 3D shape. 3D shape. So yes. And the quaternary one, basically, you already know the hemoglobin, right? So it is the, yeah, arrangement of multiple polypeptide chains. There are many, many proteins, they are linked together directly. Multiple polypeptide chains. Okay, and they are basically, you know, combined to form a functional protein. Functional protein, for example, you already know the hemoglobin. The hemoglobin. That is something you have to remember about the structure, the four levels of structure of proteins, the primary, secondary, tertiary, and the linear sequence. Basically, they're joined together by these peptide bonds, right? Now, if we talk about the functions of proteins, guys, you already know the very important function, it is taking part in the structure, right? For example, you already know a protein that is called as collagen. Okay, it provides strength and support to all the cells of the like body. For example, type 1 collagen. I've always asked students to remember 1. So it is found in bone. Bone also contains 1. Type 1 collagen, it is found in bone. Again, a very important MCQ that they can ask. Even type 2 collagen, it is found in cartilage. I'm knowingly writing the spelling of the cartilage wrong just to indicate all the cartilages of the body. They have type 2 collagen. Collagen is nothing but a protein. So it is giving strength to all the like various parts of the body, right? Even if you want to remember, guys, we have type 4 collagen. Type 4. 4, you have to remember 4 under the floor. Under the floor, it is basement. All the basement membrane of the tissues, basement membrane, they will have type 4 collagen. They will have type 4 collagen, right? Type 3, it is found in various blood vessels, the arteries, right? Again, so the most important one, it is helping in the structural formation. Secondly, for example, if we have taken hemoglobin, guys, now what is hemoglobin doing? Second function is transport. So the hemoglobin, it is basically involved in transport of oxygen, right? Hemoglobin, it is involved in transport of oxygen in the blood, right? So yes, even we have membrane proteins, like they help in transport of molecules inside and outside the cell, right? So that is something you have to remember. Even for the muscular contraction, for the muscle contraction, we need two proteins that you have already studied in class 11, the actin and myosin. Sliding filament theory, the actin and myosin, these are also proteins, right? The proteins will also act as receptors. They will also act as hormones, right? So they are helping in signal transduction pathways, right? So this is something in short you can just write about proteins also. The fourth one that they have included in the question, it is the nucleic acids. These are the nucleic acids that you have to remember. The fourth one, that is the nucleic acids, and you already know the nucleic acids. They are basically made up of three things, right? They are made up of three things. We have the base. If we talk about the structure of the nucleic acids, it is the base. Base is nothing but the nitrogenous base. We have the pentose sugar and the phosphate So we have the base, the nitrogenous base, the pentose sugar, and the phosphate, right? So the nitrogenous base, they are again classified into purines and pyrimidines, right? The purines and pyrimidines. Purines, you already know, we already know, learn by the mnemonic, all girls are pure. All girls are pure. Pure means purines. A means adenine and guanine. Pyrimidines are cytosine, thymine, and uracil. Uracil, it is found only in RNA. Thymine, it is found only in DNA, right? So remember, guys, the nucleic acids, the pentose sugar, we already know we have deoxyribose and ribose sugar. Deoxyribose and ribose. Deoxyribose, it is found in DNA. Ribose, it is found in RNA. Okay, so we have deoxyribonucleic acid. Nucleic acids are again of two types, DNA and RNA. DNA stands for deoxyribonucleic acid, and RNA stands for ribose nucleic acid. Ribose nucleic acid, right? So DNA, basically it is double standard. It is, and RNA single standard, right? So RNA is basically involved in protein synthesis, right? Now, if we talk about function in short, guys, function you should remember. If we talk about DNA, basically it stores the genetic information. It stores the genetic information, and you already know it is passed from the parent to the offspring, right? It stores the genetic information, and this genetic information is passed from the parents to the offspring, right? Now, secondly, the protein synthesis, the RNA, guys, you already know it is involved in the protein synthesis, the central dogma of molecular biology. From DNA to mRNA, it is, yes, let me just write it down. From DNA to DNA, it is replication. From DNA to mRNA, it is transcription. And from mRNA to proteins, it is translation. So yes, the RNAs are basically involved in the protein synthesis, right? So the major ones you have to remember. Even the RNAs are also like they try to know or regulate the gene expression, right? So they also help in regulating the gene expression, regulating gene expressions such as the microRNAs, right? We have discussed this in earlier classes also. Okay, even the long non-coding RNAs, they are also no regulating the gene expression. So that is the first answer that you have to just write. Okay, it's a 10-mark question, guys. So yes, obviously, the question says you have to mention the structure, you have to mention the function of all the four biomolecules, the carbohydrates, the lipids, the proteins, and the nucleic acids. So first, if we are taking carbohydrates, try to draw the structure if you can draw, right? And try to mention the function that we have discussed, right? The lipids, right? The simple lipids compound derives how, what is the structure of simple lipids? They are just simple fatty acids and glycerol. The compound lipids, we have fatty acid, glycerol, and some other compounds like phosphate, or if it is carbohydrate, or if it is sulfate, right? If we have derived lipids, so the triacylglycerol is broken down to release no fatty acids and glycerol. These are again the derived lipids. The functions of lipids, right? The phospholipids, the hormone, the hormonal function, the energy storage. The third, it is the proteins that we have discussed. So we have four levels of structure: the primary, secondary, tertiary, and quaternary structure of protein. So you have to mention about that, and we will be taking up this separately also because we have another like question, and the functions of proteins, right? And the nucleic acids. So basically you have to tell the structure, the base, the pentose, sugar, and phosphate, or even you can draw that structure, but it will be very time consuming. So just try on to mention it in the form of a flowchart that I have drawn. Okay, just try to mention like this and just try to mention the function. Try to give broad headings over there and try to highlight the important keywords, right? So that is again a very useful tip that you can just follow for this particular answer that is of 10 marks, right? So this question is done. Now coming on to the next question, guys. Now the next question says, describe the structure and properties of amino acids and their role in protein synthesis. And their role in protein synthesis. Now it is a short answer type of a question. In the starting only I have told you. Either it can be asked like 8 marks or 5 marks. More like probability is like it will be asked in the 5 marks fall part, right? So we are going to discuss anyways the entire topic, and I will be highlighting the important ones that you have to mention in the exams. Now, you already know the amino acids. The amino acids, they are basically building block of proteins. They are building block of proteins. And remember that the proteins or the amino acids, they are coded by the genes. They are coded by the genes. We already know they are the workhorses of genes. Workhorses of genes, right? Now, firstly, just try to read the question and try to incorporate what is there in the question. The question says the structure of the amino acid. So just try to draw the structure first. Structure of the amino acid. Structure of amino acid. Now, we have a carbon over here, the alpha carbon atom, which is occupied by 4 different valencies. All the valencies we already know. This carbon will be having 4 different valencies. That's why it is the alpha carbon atom. The alpha carbon atom. Now, at one end, we will have the NH2 group. NH2 group, that is the amino group. Remember, guys, this is the amino group. The base group, which is over here, the amino group, right? It is a basic group that can accept a proton, okay? So it is contributing to the basic structure of the amino acid. On the other hand, we have this, yes, the carboxylic acid group, right? The COH group, it is the acidic group. It is the acidic group that can donate a proton. We have the hydrogen atom. We have the hydrogen atom, right? It is attached to the central carbon, okay, the alpha carbon atom, and the side chain, that is R group. Now, when I say R group, it is basically referring to the number of carbon atoms, the number of carbon atoms, the number of carbon atoms can vary. The number of carbon atoms can vary, right? The number of carbon atoms can vary, right? So yes, that is something you have to remember about the structure of this amino acid. Now they're asking the properties of the amino acids, right? Properties of the amino acid. Now, okay. First is the structure done, then the properties of the amino acid and role in protein synthesis. Now let us just try to answer the second part of the question, that is the properties of amino acids. Properties of amino acids. Now, firstly, remember guys, they can exhibit isomerism. Exhibit isomerism. Now, when I say isomerism, guys, now let me try to draw the structure for you people. They can exhibit isomerism. Now, let us suppose this is the mirror over here, right? And I am trying to draw a very basic alanine. That is one of the amino acids, right? Alanine. So we already know we will have one hydrogen over here. We will have COOH group, and just like it's donating, yeah, COO minus. We have, yes, the R group, that is CH3 over here. And over here, we have, let us suppose, NH3 plus, right? NH2 is basically accepting, yeah. So yeah, it is NH3 plus. Now we just try to draw, this is called as D-alanine. The mirror image, guys, the mirror image, it is, yes, it will be like simply like this. Hydrogen will be over here and it will be CH3. It is called as L-alanine. Now, this carbon atom, it is called as the asymmetric carbon atom. Now, when I say asymmetric carbon atom, it means that all four valencies, they are basically occupied by different carbon atoms. They are occupied by different carbon atoms. I mean, different groups actually, different groups. They are occupied by different groups. Obviously, you can see we have NH3 over here, we have CO minus, we have H, and we have CH3. Now, guys, remember most of the amino acids in our body, they basically exist in L form. Very, very important. They exist in L form, right? So again, guys, there is an exception which does not all the amino acids, all the 20 amino acids will exhibit the isomerism except glycine. Glycine, basically, it is also an amino acid, but there is no asymmetric carbon atom. There is no asymmetric carbon atom, no optically active carbon atom, or I can say no chiral carbon atom, right? Or there is no D or L glycine. Only glycine is possible, right? Because yeah, we have H only, right? Now, L amino acid can be converted directly to D amino acid, okay, by the help of enzyme called as racemase, right? So it can act on both D2L and L2D, right? So that is something which you have to remember. Again, two important questions that they can ask in the MCQ wala part, which is from here, which you can take that is D-serine and D-aspartic acid. Very, very important. D-serine and D-aspartic acid, they are present in brain. Most of the amino acids in our body, they exist in the L form, except for the two, that is the D-serine and D-aspartic acid, they are present in brain, right? So remember, that is something you have to remember. That is the first property of this amino acid. Secondly, guys, you have to remember the amino acids exist in different charged states. Different charged states. Okay. So, yes, based on two properties, guys, over here, based on two properties that you have to remember. We have first that is called as the pH of the medium and the isoelectric pH. Isoelectric pH, that is PI. Isoelectric pH, that is PI. So we can have like three situations, guys, based on this. We have at pH is equal to isoelectric pH, isoelectric pH, at pH more than isoelectric pH, and at pH less than isoelectric pH. So we have like three situations that you have to remember, right? Now for every amino acid, guys, there is a point that is called as the isoelectric point, right? So yes, if we have the positive charge equal to negative charge. Now guys, if at pH is equal to isoelectric pH. Now guys, just try to remember, if we have one amino acid containing this, okay, and another, yeah, if we just try to, yeah, make it in charge form, it is COO minus and NH3 plus. So you can see in this amino acid, the positive charge, it is equal to negative charge. Positive is equal to negative charge, so that there is no net charge, neutral charge. These are called as Zwitter ions. These are called as Zwitter ions. Okay, in the same, you have positive charge also and negative charge also in the same amino acid. So obviously there is no net charge. It is called as neutral charge or the Zwitter ion, right? Now remember, guys, there are some more properties, guys, of amino acids. There is no mobility in electric field. No mobility in electric field, right? There is no net charge. Obviously, if the net charge is zero, there is no mobility in electric field. There is maximum precipitability, precipitability, right? There is minimum solubility, and there is least buffering action. There is least buffering action, right? There is least buffering action, right? Now, we have discussed like two of over here. They exhibit isomerism. They exist in different charge state. One more, guys, which you can remember over here. The third one, the amino acids. Basically, they take part in buffering action. Buffering action. Now, buffers are something, the solutions that resist the change in pH. Okay, if there is if there is a small change in pH, these basically try to maintain the pH of the blood. Okay, so yes, we already know the Henderson's equation, right? Hasselbalch equation, right? So that is we have discussed this in that part also. And one more that you can remember, that amino acids absorbs UV light, right? Now, remember, guys, that amino acids, basically they do not absorb visible light. Okay, that is why they are colorless. That is why they are colorless. But these amino acids can absorb UV light. And this graph has been asked in the MCQ. Again, a very important part of MCQ that I'm discussing over here. Yes. So if a graph is given like this is for tryptophan. Tryptophan. Then we have tyrosine, tyrosine, and yes, we have phenylalanine, phenylalanine, right? So tryptophan is one of the amino acid that is having maximum absorption. Very, very important question for MCQ also, for Viva also. Maximum absorption, it is by tryptophan, right? So basically tryptophan, tyrosine and phenylalanine, I'm basically talking about, these are the aromatic, the one which are having the cyclic structure. They are aromatic amino acids, okay, because of conjugated rings, right? So basically, they absorb UV light in the range of 250 to 290 nanometer. Again, very, very important MCQ. They absorb UV light in the range of 250 to 290 nanometer, right? So 280 is like, if 290 not given, 280 is basically, yeah, it's the range of maximum absorption. It's the range of maximum absorption. So these are like the four points that you can just remember, right? The properties of amino acids, okay? And the role in protein synthesis. What is the role of amino acids in protein synthesis? Role of amino acids in protein synthesis. Now, one thing that you already know, from DNA, mRNA is formed, and that is by the process of transcription. And from this mRNA, what is formed? Proteins, by the help of translation, right? Right? So that is you have to remember. So basically the mRNA, there are basically the codons, right? We have the codons, the three codons, that is the three nucleotide sequences. Okay, they are read by each ribosomes. Now these codons basically, okay, each codon basically there is corresponds to a specific amino acid. Okay. Now we have something called as tRNA, that is the transfer RNA, which actually brings the appropriate amino acids to the ribosome, right? So yes, again, one is the protein, okay, formation, right? That is the important part of these amino acids, right? Secondly, yeah, this is like, you can just remember, you can include this in the answer. That is like more than sufficient for 5 marks question. Right? So that is the structure and properties of amino acids and the role in protein synthesis, right? Now, coming on to the next question, that is the third one for the today. Explain the different types of lipid molecules and their significance in cellular structures such as membrane. Now, different types of lipid molecules. Question number, okay, this is 4. We have discussed question number 2 done, question number 1, question number 2 done, question number 3. Now, guys, you already know. First is just the, okay, we have the simple lipids, the Bloor's classification, the simple lipids. Now, when I say simple lipids, they are nothing but the esters of fatty acid and alcohol. Alcohol is nothing but the glycerol, right? Or any other higher. Most commonly, it is the glycerol, or it can be any other higher alcohol, right? So simple lipids are nothing but fatty acid plus glycerol. For example, fats or oils, right? Now fats, when I say fats, it is triacylglycerol. Earlier, no, it was triglycerides, so they are solid at room temperature. Oil, basically, it is liquid at room temperature, right? We have waxes also. We have fatty acids plus high molecular weight alcohol, right? For example, bee wax, right? So if we talk about the, yeah, now, secondly, you have to remember the complex lipids. Complex lipids are nothing but fatty acid plus alcohol plus other group. Fatty acid plus alcohol plus other group. Fatty acid is basically, you already know, if the other group is phosphate, okay, it is called phospholipids. It is called as phospholipids. Phospholipids are the ones which are very, very important in the structure of the membrane, right? If it is carbohydrates, it is glycolipids. If it is sulfate, it is sulfolipids. If it is proteins, it is lipoprotein. It is lipoprotein, right? So we have different lipids, right? They are involved in different parts, right? So yes, my class later. Sorry. Okay. So these are the complex lipids. We have derived lipids. Derived lipids. Now, guys, phospholipids are basically arranged in the form of lipid bilayer. Lipid bilayer. They're composed of head and the tail part. Okay. The head being hydrophilic, they are more towards the water wala part, and the tail being hydrophobic. Hydrophobic. So they are arranged like the lipid bilayer, the tails inside and the heads like outside, right? So you can just see that the head are facing the extracellular fluid and the internal heads which are there, the inner row, it is basically facing the intracellular fluid. So they are basically arranged in this format and they are, you already have read about the Singer and Nicholson model, right? So that is again, you can just remember over here, right? Derived lipids, you already know the ones, they are formed from simple or complex lipids. They are derived from simple or complex lipids. They are derived from simple or complex lipids. For example, um, triacylglycerol, when it is broken down by the help of enzyme lipase, it is broken down into three fatty acids plus glycerol. Now, this three fatty acid and glycerol, they are nothing but the derived lipids only. Examples of derived lipids can be the examples of derived lipids can be fat soluble vitamins. We can have hormones. We can have steroids. We can have ketone bodies, right? These are all derived lipids, right? So that is something you have to remember. Okay. Yeah. Now I have already spoken about the phospholipid, right? One important thing you can also remember, that is the glycolipids, guys, right? The glycolipids, they are similar to phospholipids, but they are actually having the glyco component, that is the carbohydrate, right? So they are attached to the hydrophilic head. Now, if we already know, if it is the head part and this is the tail part and this is structure is of a phospholipid. So basically, these glycolipids are basically attached to the head part of the phospholipid, right? So basically, glyco means the carbohydrate group, right? So yes, what is the function of these glycolipids? They help in cell recognition and signaling, right? So yes, that is, you can just remember. Okay. So yeah, that was regarding the different types of lipids, right? And their structure, role in the cell membrane. Now outline the different levels of protein structure, guys, from primary to quaternary levels. Question number 4, I suppose. Yeah. Now, we have primary structure, secondary structure, tertiary structure, and quaternary structure. Remember, guys, now when I say primary structure, guys, the primary structure of proteins, they are basically referring to the linear sequence of amino acids. Linear sequence of amino acids. linked by peptide bonds. Linked by peptide bonds, right? They're linked by peptide bonds, right? So yes, example you have to mention, that is the insulin. Insulin, yes, that is composed of 51 amino acids arranged in a linear fashion. Obviously, if this is right, you already know. I'm just talking about, yeah. Now, guys, just try to remember, you can just remove this water molecule. There are two amino acids. There are two different amino acids, amino acid 1 and amino acid 2. Again, there is one separate question, but I'm discussing it over here only. So if there is a loss of water molecule, okay, then obviously this C will be bonded to O and this N will be kept with H only, right? This bond, C double bond O N H, this is called as the peptide bond. This is called as the peptide bond. They are linked together by the peptide bond, right? For example, insulin, right? Then we have the secondary structure of proteins. Secondary structure of proteins. So basically refer to the folding of the proteins, right? Folding within a polypeptide chain, within a polypeptide chain. Okay. Now the folding is basically stabilized by hydrogen bond. It is stabilized by hydrogen bond, right? So yes, it's like between the central atoms, not the side chains actually, right? So basically we have alpha helix and beta sheath, right? So alpha helix, it is basically a right-handed colloid structure, right? So it's formed by the hydrogen bonds, right? And beta sheath, it's basically they run parallel or anti-parallel to the to each other, right? So that just like the plates of the sari, right? So yeah. Example, you can just remember keratin, right? The keratin, basically the protein which is found in the hair and nails, right? So they contain a very, very high amount of these alpha helices. Okay. Even the silk, guys, the silk, it contains the beta sheets. Tertiary structure. Tertiary structure, it is basically the three-dimensional folding of single polypeptide chain. Of single polypeptide chain, okay, into a final shape, right? So basically there are various interaction, guys, we have already discussed in the regular classes, no? The hydrophobic interactions, the hydrogen bonds, the disulfide bridges, right? We have already discussed the disulfide bridges also, right? The example of tertiary structure, it is myoglobin. So myoglobin is basically a oxygen binding protein, right? It's an oxygen binding protein, right? It has a tertiary structure and no, it helps to bind the oxygen and helps to release the oxygen efficiently. Okay, we have quaternary structure, the quaternary structure, and that is something you already know. It is the arrangement and interaction of multiple polypeptide chains. Multiple polypeptide chains. Okay, that is the subunits, right? Into a functional protein complex, right? So yes, that example is the hemoglobin, right? The oxygen carrying protein in the red blood cells, right? So it consists of four subunits. You already know the structure of hemoglobin consists of four subunits, two alpha and two beta. Okay. And they take in the oxygen, bind to the oxygen and release the oxygen. Right. So that is again regarding the, yes, protein structures, which we have discussed. Essential amino acids are the ones, guys, again, it's a very short kind of an answer, right? The fifth answer. So remember that the body, essential amino acids, the body cannot synthesize on its own. Okay, they must be obtained through diet. They must be obtained through diet. They must be obtained through diet. Okay, they must be obtained through diet. Okay, just yeah. What? Uh, hmm. They must be obtained through diet, guys, and you already know, based on nutritional requirement, basically we have eight essential amino acids. Remember, it is methionine, it is threonine, tryptophan, all the branched chain amino acids, guys, you already know, the valine, isoleucine, and leucine, even the phenylalanine and lysine, they are also very, very important. So we have the eight yes, essential amino acids that should be supplied via diet. Describe the term peptide bond. Yes, I have already discussed this. The two amino acids, they are joined together by the help removal of a water molecule, and we have just discussed this. Yes. Significance of nucleotides in energy no transfer process. Now over here, guys, just try to mention regarding the very first one, that is ATP, adenosine triphosphate. Adenosine triphosphate. So it is known as the energy currency of the cell. Energy currency of the cells, right? So yes, the molecular unit of currency. So it consists of ATP. What is the sugar, guys, over here? Right? The ribose sugar. We have the base, the nitrogenous base is adenine, right? And we have the three phosphate molecules, right? So yes, the energy in the ATP, basically it is stabilized or it is stored as no high energy phospho-anhydride bonds, right? Between the phosphate groups, between the three phosphate group, we have the phospho anhydride bonds. Now these bonds are high in energy because they are negatively charged phosphate molecules, right? So yes, the ATP can be broken down into ADP, adenosine diphosphate, and PI, that is one phosphate will be released for no energy, right? So yes, if the ATP is further broken down, it can be released. It can be converted into adenosine monophosphate, and two phosphate molecules can be released. So if only one smaller amount of energy, if more than two phosphates are released, it means high amount of energy. So yes, ATP, you already know it's used in various energy processes, demanding processes. For example, the very common, that is the muscle contraction. The muscle contraction, the active transport. Yes, the transport that you have studied, that takes place by the use of these ATP molecules, right? Even the cell division requires ATP, right? Again, we also have, yes, the other one, that is GTP, guanosine triphosphate. Guanosine triphosphate, remember it is similar to ATP, but over the nitrogenous base over here will be guanine. Okay. So it also contains high amount of these phosphate bonds. And remember, GTP is useful in signal transduction. Okay. Signal transduction, for example, we have a receptor called as G protein coupled receptor. G protein coupled receptor. Okay, so G proteins act as molecular synthesis, right, in response to signals from outside the cell. So that is something you can remember. You can also write about cyclic AMP over here, right? Cyclic AMP, the secondary messengers and all, right? So that is regarding about the today's session. So yes. Thank you. And these are the various social media handles which you can follow for the updates of the upcoming classes. Thank you.